Yeah, but please interrupt me with questions and anything you want clarified in particular. Again, I'm not going to assume knowledge about fMRI, but that doesn't mean I won't err. Okay, so first of all, I want to start with a land acknowledgement and acknowledge that I work and live on the unceded ancestral traditional territory of the Musqueam Nation. And there's something in the chat already. So this is also, okay, Catherine is saying I'm other ADHD presentations. This chat is dangerous in front of me. Okay. So one thing I sort of have been increasingly aware of is the importance of studying brain activation in healthy humans in the context of translational research. And often when I'm in situations where people are talking about translational research, they're like, oh, well, we'll figure out all this stuff in the rodents, like we'll do opto and chemo and activate and inactivate circles and receptors, and we'll give them drugs and we'll know all the circuitry, and then we'll just go into the clinic and we'll figure stuff out. And I was actually presenting something related to this mental health and addictions IRP to the scientific board of advisors of the Center for Brain Health. And some man who I feel like so warm and fuzzy about said, but wait, it's really important to do preclinical work in quote unquote healthy, relatively healthy, not clinically referred humans, because rat brains are not human brains. I mean, we often try to look at processes that we think are fairly conserved or are conserved, but they're still very different. You can't assume that what you see in a rat is what you're going to see in a person. And the methods we use to look at them, and this is kind of me kind of highlighting it, are really different. Like rodent researchers can activate and inactivate. They can infer causality. They can target specific circuits, right? They can turn stuff on and off as well as damp it down and ramp it up. Or they can cut it out, right? Just cut it out. Often when we're using fMRI, what we are doing is just measuring the BOLD response, the blood oxygen level dependent response, which is a measure of the magnetic properties that differ between more and less oxygenated blood. So we're looking at like differences in the activity of pretty much everything that is in that often 3 by 3 by 3 millimeter hunk of real estate, including all kinds of types of neurons, including glial cells, anything that needs to metabolize. We're seeing its activity. So it's really different. So the thought that like you can sort of manipulate a region or a circuit in a rodent and that will translate into BOLD activation, which is also very slow, it doesn't necessarily follow. They're just, it's very apples and oranges. So my argument that you need human preclinical research as well as rodent preclinical research. So, you know, you need to, so a couple of things, again, that came up in this meeting. First of all, you know, okay, so you're seeing, you know, this pattern of the nucleus accumbens and pre and infralimbic cortex in rodents related to this particular behavior. But you need to verify that those regions are actually active in humans in the same behavior. So that's number one. But number two, then when you're doing human brain imaging, instead of having it be a really poor person's version of what rodent researchers could use, you know, you can actually leverage what we can do to start modeling stuff about the representational content of the activation in these sort of large bits of the brain. And I think that's where we can sort of, instead of just confirming the rodent research, we can actually start to add, elaborate on it. And in doing so, you know, in both cases, provide a ground truth for clinical studies. So I just like this slide. So our lab is doing a couple of translational studies. We started with a study I'm about to show you about, which is a collaboration with Stan Floresco and Trisha Chakrabarty in the Mood Disorders Clinic, but we also now have a collaboration starting with Kath's lab, and it's a way that I find it, you know, I really like being the filling in the translational sandwich that I'm about to illustrate. So, as I mentioned, Stan can go into his rodents and he can activate, deactivate, lesion, opto, chemo, give drugs. He can sort of learn a lot about the circuitry and pharmacology of a certain behavior by doing that. And then you have in the clinic, you have Tricia, who's a psychiatrist who is seeing patients with major depression. And she is trying to figure out what to prescribe them. And she wonders, she struggles with the idea that like, well, I don't know who's going to benefit from what. And also the fact that the sort of the subtypes of major depression are quite under, it's well known that, for example, that often people who are with high levels of depression have high levels of anxiety. But how people with high depression, high anxiety may differ from people who just have high levels of one or the other, not to mention neither, is something that's been kind of under researched. So that's a real interest for Tricia. So I argue that we are the meat in the translational sandwich, and what we can do and have done is we take a study that Stan has used in rodents and knows, a lot about the circuitry underlying certain behaviors. We can then back translate it, show it to a whole bunch of humans online because COVID, and see if in this large population of people with a range of anxiety and depression scores, certain patterns of behavior maybe are related to having high levels of anxiety and depression versus high levels of one and not the other. And what we can also do is use fMRI to see whether we do sort of recapitulate similar patterns to what Stan has observed in his rodents. And also we can elaborate on it, which is what I'm going to talk about in this talk. And then finally, what has happened so far is we found a particular pattern of behavior that was related to particular subtypes of high anxiety and high depression, which then Stan went back and gave some yohimbine to his rats and some other drugs to his rats, and found that actually this pattern of behavior was related to norepinephrine activity, which is cool. So if that bears out, and we're also going to the clinic, and Tricia's on mat leave right now, so we're not doing it, but we're sort of partway through a study where we're seeing if these patterns are also observed in clinical populations with major depression and with subtypes of major depression. And so, ideally, we go on, if Kath and I have our way, some decades from now, to actually be able to give pharmacological challenges to people. And eventually Tricia will have a better idea of what to prescribe her patients to reduce their suffering. So the sort of the example of the study, this study is we're looking at specific avoidance behaviors and to see what characterizes subtypes of depression and what it means for treatment. Okay, but the fMRI part is what I'm going to talk about today. And I'm going to use an example from a different study, a previous study where we kind of developed this approach to looking at fMRI. And then I'll talk about a little preliminary work we've done with some pilot data from this particular study. And again, this is where I want to spitball. Okay, how can we kind of use this approach in a translational context in particular? Because although the study I'm going to tell you about was not explicitly a translational study, it actually could have been, because it was a very simple Pavlovian learning task, so easily. So the approach that was developed in my lab has the potential to model the kind of informational content that's being carried by the brain during a particular task or behavior. So I'm gonna outline an example of the methodological work of James Crickleway. He, until this past summer, was a postdoc in my lab. Now he is an assistant professor at Lakehead University, who's also, just for fun, teaching full-time at UBC this term. I know. So I'm going to talk about the approach which we call a pattern of interest approach that uses representational similarity analysis, and I'm going to define those terms. But representational similarity analysis, or RSA, is a multivariate approach to analyzing fMRI data. It's been around for more than 15 years now. And again, I'm going to review it in a minute. And what Jamie innovated in parallel with people in a couple of other labs who sort of developed parallel approaches is a way of using RSA to test models of the informational content that's instantiated by different large-scale brain regions during experimental tasks. And so that part we've deployed pretty successfully over the past couple of years. The proof of concept paper just came out in eNeuro this January. And the second proof of concept paper is under revision for JNeuro. So we're slowly making progress. So representational similarity analysis, I'm going to start by reviewing what that is. So if you're an fMRI person, you are probably familiar with it, but if you're not an fMRI person, you may not be. So this was introduced by Nico Kriegeskorta. I'm sorry, Laura, I'm probably mispronouncing his name horribly. How do you pronounce it? Oh, okay, I wasn't too far off. Kriegeskorta. So this is a mind reading approach where you read in from patterns across voxels the degree to which the brain is instantiating information about stimuli or conditions as being more or less like or unlike each other. So what is a voxel? A voxel is this grid we impose when we do an fMRI scan, a functional magnetic resonance imaging scan, we impose a grid on the brain. And that grid is voxels, or three-dimensional pixels, which are these little cubes. They may be 2 by 2 by 2 millimeters or 3 by 3 by 3 millimeters. So big, if you're thinking in terms of neurons, very big. And they don't map on to the architecture of the brain. It's just a grid we impose on the brain. And then we extract the bold response, the ratio of, of the magnetic properties of the ratio of oxygenated to deoxygenated blood from every one of those cubes. Okay, so those are voxels. Then in sort of a basic RSA analysis, we extract activation from the voxel, we do some pre-processing on it, and we run a regression on every voxel. So these little squares here are just little schematics of where each small square is a voxel, two-dimensional depiction of a voxel. And for each of these, we have run a regression and gotten a beta value out of it. So these little checkerboard patterns are patterns reflecting the beta values, reflecting more or less bold activation. So we'll say the bright yellow is a lot of bold activation and dark red is not very much, relatively a lot less bold activation. So you might show people in this example, which is from this original Dr. K paper, you would extract the activation, choose a region of interest, and let's say here it's a region, the fusiform face area. And you look at the pattern across voxels of more or less activation as reflected by the beta weights for this presentation of the house or the presentation of this house. And then you look at similar voxel patterns between the presentation of more houses and also between the presentation of faces. And then what RSA does is correlate these voxel patterns, these patterns of more or less activation across voxels. So it correlates this with this, and this with that, and this with that, et cetera, to which you have the correlation matrix. And to make things more confusing, this may be called a dissimilarity matrix or a similarity matrix. And they're, of course, just flip sides of the same thing. But this is from the original paper, and it's a dissimilarity matrix. And so these are just correlations. So this scale here says it's dissimilarity, but basically that's negative correlation, right? Bright red is a more negative correlation, and dark blue is a positive correlation, right? So these two faces, it's the same face, the same trial. So they have zero dissimilarity. The patterns are identical. But of course, this face shows quite a bit of dissimilarity with this house, right? And then what you can do with RSA is you can then take these correlation matrices and you can visualize the same information in the similarity graph icon, and you can even do it sort of with some mathematical precision. You can put this into Euclidean distance. And so you can see that, and then people talk about representational space. So in representational space, you have, two faces that look quite a lot like each other and quite a lot unlike houses. So again, and the distance of the lines is really the amount of the correlation. And houses look like each other, and houses don't look like faces because were in the fusiform face area or the parahippocampal place area. And again, what the original paper sort of proposed is you can then choose regions. This is throughout the ventral visual stream, and you can choose regions and see how different regions are representing these stimuli in representational space is more or less similar. So here you have the fusiform face area and the parahippocampal place area, and you see this pattern we just looked at where faces look like faces and houses look like houses. But if you go up to the anterior temporal lobe here in a region that is maybe sensitive to individual face identities, then you have these sort of the male and the female face quite distant in representational space. So this is representational similarity analysis. So it was the first step to what sort of Jamie did in these studies. So this is now an example study. I don't want to spend a lot of time like, ooh, here's what we found and what we didn't find, because I'm really using it as a way of illustrating this approach that I think may be useful in translational research. And again, that's what I kind of want to spitball. So this is a very, very simple Pavlovian learning study that used affective touch. So it used pressure pain or a soft brush stroke in the scanner. And really the question that we were asking in this study is how is the affective versus sensory information represented and learned, in particular hunks of real estate of the brain. So basically the question was, when we learn to associate cues with pain or pleasure, what information do we attach to the cues? Is it sensory information, or is it the emotional associations of goodness and badness that go with it? And so the challenge here was to see if we could read the brain to see what information was being carried when they were actually being touched, the unconditioned stimulus, the US, when they were getting the pressure pain or the soft brush. So during the experience, and then when they just saw the cue that had come to be associated with it before and after Pavlovian learning. So this is just, these are Jamie's animations, and Jamie was on the job market, so he did some really nice animation of his slides. So some background on affective touch and why the touch system was actually a cool medium for examining these questions. So you whack somebody's hand. We actually didn't use a hammer. We used actually a pressure pain device, but you whack somebody's hand with a hammer. You have one family of receptors that carry information along a fast myelinated afferent line. And these are pain-specific receptors, right? And then it runs quickly to the, no, sorry, these are not pain-specific receptors. Take it back. These are discriminatory receptors. So they carry information along a fast afferent line that's myelinated, and it runs quickly to somatosensory cortex with information about the quality of the touch. Maybe what makes it hammer-like, metallic, cool, heavy, high pressure. And then these are the pain receptors. You have another family of receptors, and the information runs more slowly along these unmyelinated pathways, and it pitches up in the insula and ventromedial prefrontal cortex and anterior cingulate cortex. And this carries more information about the affective qualities of pain. Okay, but again, in this study, what if you're not directly hit by a hammer, but you see a face that is associated, that you have learned has come to predict what happens. It comes to predict that you're going to be hit with a hammer. So then what information do you represent? Is it the information carried by the lines about discriminatory touch, affective touch, both? And that was what we kind of wanted to represent. And the nice thing too about the affective touch system is there is an equivalent affiliative touch system. So it's responsive to 3 centimeter per second soft stroking touch, only found on the hairy skin. And it too has sort of valence specific unmyelinated fibers that pitch up in pretty much the same place as the pain fibers that are similar, as well as the discriminatory touch receptors going into S1. Okay, so this is a simple Pavlovian study. Faces came to be associated with painful and pleasurable touch or not. So just an illustration. Again, thank you, Jamie. Here's the voxels in the brain. And here are time series of the bold response for each voxel. And again, in this task, we wanted to measure the bold activation after they felt the painful. or pleasant touch. And we wanted to read out that information that was being instantiated in the patterns across voxels. And then a second part was to use that to try to predict the activation patterns when they saw the face that was associated with both the painful and the pleasurable touch in the absence of actual touch. So here is a super, super simple experimental design. So there were acquisition blocks in which faces were paired with either soft brush in this case, but there were other blocks where they would be paired with pressure pain, actually an entire sort of separate task where they were paired with pressure pain. And there were actually two faces that were paired. So they were the CS plus, and then there was one face that was never paired, and that was the CS minus. And then we also had blocks where they only saw the faces. So only the CS plus and CS minus. And importantly, we wanted to have one of these blocks first. So we wanted to be able to see what their neural responses were to the faces before they'd ever been paired, as well as after they'd been paired. Okay, so Jamie's approach is a form of pattern component modeling. And again, pattern component modeling is where you model sort of idealized patterns of the correlation patterns that you would see if this brain region was only representing one bit of information. And so PCM was proposed by Nico in 2013, and then sort of in parallel, a whole bunch of people started working on it, including us. A lot of the approaches are very data-driven. Ours is pretty hypothesis-driven. So Jamie and the rest of the lab came up with some patterns we thought should be represented under different circumstances in this task. So this is if a part of the brain is only representing painful touch, then these would be the painful touch trials, and this would be a correlation of 1. They would be perfectly correlated with each other and absolutely not, zero correlation with anything else. And the same if it was only independently representing pleasurable touch, then the pleasurable touch trials would be perfectly correlated with other pleasurable touch trials and with nothing else. And then this is just what it would look like if it was valenced. So if pain trials were perfectly correlated with each other, but perfectly anti-correlated with pleasure trials. So these are patterns of interest or ideal similarity matrices that we're going to use to model activation. Yeah.
Sorry, so I'm just trying to find the valence touch bit. So,
yeah, I think this is a flawed image too, by the way.
Is this a dissimilar?
So this is a similarity matrix. So when we move over to our data, we're flipping from dissimilarity, sorry about that, to similarity. So red is a correlation of 1 and blue is a correlation of -1.
And this would all the squares voxels?
The squares are now, they're the correlation matrices and the dissimilarity matrices. This is what gets so confusing, is you have all those checkerboard patterns, but it's this, only it's flipped. This is a dissimilarity matrix. Now I'm showing you an idealized similarity matrix.
Okay, I've got it.
Yeah, that gets me still. I'm like, wait, what are the checkerboards I'm looking at? So yeah, these are similarity matrices now, idealized. Okay, and this is another wonderful Jamie animation. This is an example of how these pattern of interests were first applied to model the trials that contained actual touch stimulation, the US, in order to examine the degree to which a given brain region represented sensory versus hedonic and other information. So again, here's the hand that's being affected. Here's S1. Here's the ventromedial prefrontal cortex. Okay, and this is going to be now the actual data that we observe. These are two similarity matrices, one for S1 and one for VMPSC. Here are trials in which there was pressure pain. This is a correlation scale. So in this particular one, in this similarity matrix, a perfect correlation would be dark red, and a perfect negative correlation would be blue. And these, again, are all the trials where pressure pain was involved. So again, they're seeing a face and actually feeling the pain in this case. And here's the VMPFC. Oh, no, sorry. And here is the trials in which they experienced soft brush stroke. Okay. And now here's the trials in which they were not touched at all, the CS minus trials. So no touch. And that may be the pattern of data we actually observed. And that's representational similarity analysis then tells us, you know, what trials are more distant from others in representational space, right? So that's what we've got. So then pattern component modeling is the next layer. And again, this is Jamie's bit. So he's created these ideal components. So here's just modeling pressure pain for the VMPFC, just modeling pleasant brush. And then there was a, oops, sorry. So that's for the VMPFC. And in order to find out, so basically he used regressions. He used this greedy best first search algorithm to iterate through combinations of these ideal POIs to see which combination of POIs predicted the actual data to the point that we could reconstruct the data. And so this is pretty much, so then for S1, you don't expect it to be so much about the pain or the pleasure, but specific touch type meant that either there was pain or there was painful touch, there was pleasant brush, or there was no specific touch, right? It's like just lying in the scanner touch, nothing to the hand. And tactile manipulation, which just meant there was either pressure or pain, there was active touch, pressure or pain, but it didn't care whether it didn't care about the valence. Okay. And scanner block was it the pleasure block or the pain block? So basically these are all sort of, when you combine them, they reproduce the activation that we actually observed in the primary sensory cortex, and it's mostly non-valence tactile signals. And then you combine this with a control condition that I always forget what exactly it means, and combine them together to reproduce pretty precisely the actual pattern that we saw, the similarity matrix for the VMPFC, and this is sort of mostly valence labeled. So again, this is when they are actually being touched. So again, what we were sort of using that as a stepping stone towards is what's carried forward with Pavlovian learning. And so we also had these blocks where they only saw the CS faces. And so again, they saw them before and early after learning and long after learning. And you better believe in this simple, very simple Pavlovian task, our humans learned it really fast. It was single trial. You know, some of the slower ones maybe took 2, but it happened fast. I would imagine even a robot would do pretty well with that one. It wouldn't take a lot of training to get that one down. So now we're going to look at how each component of the US data predicts the activation in response to the CS at each time point. So again, early, mid, and late. So early is before there's been any association. And this is early visual cortex, which is just seemed like a good control region because we didn't really expect its pattern of activation to change with conditioning. And so this is the POI that predicted it when they were actually in the actual US condition. And experimental task, I mean, so they were all looking at faces as well as being, you know, feeling the pairings. And so they were different sets of faces. So that's probably what Human is responding to there. And then what you see, so the dark lines, so the transparent bars are the pattern that was observed from the US stimuli, and then the solid bars are the degree to which it's recapitulated when they're seeing the CS. So that's early. Whoops, what am I doing backwards? Early, mid, late. It recapitulates what happened the first time when they were looking at those different sets of faces, right? So nothing happens with learning. It's just conditioning independent reactivation. No surprise. I mean, actually, there is some evidence out there of early visual cortex being conditionable, but at this scale, not so much. So then we were interested in the reactivation of the hedonic aspects. So we looked at our friend, the ventromedial prefrontal cortex. And then what we're seeing here, the sort of POIs of interest, our pressure pain here independently, pleasant brush independently, and this weird sort of controlled temporal adjacency condition, which just had to do if they were near each other in the same block. But crucially, early, nada. Mid-conditioning, it's representing independently pressure pain and pleasant brush stroke significantly. And then a little bit later on, it drops the pleasant brush and it hangs on to the pressure pain. So although it's showing the sort of valence or each type of touch independently, each type of valence touch independently, it's not hanging on to the positive. It's dropping the positive and hanging on to the negative. And then when you get to the anterior cingulate cortex, which we know is really sensitive to pain, again, early in conditioning, nothing. Mid conditioning, pressure pain, and we're not getting anything for pleasure here. And late in conditioning, also pressure pain. So the ACC is really sort of representing information about pressure pain primarily. So the sort of, we concluded the VMPFC independently represents pain and pleasure, and the ACC represents pain. And so what we saw is the way we interpreted it more strongly when we first submitted the paper than now after the reviewers have reprimanded us. Sure. I know it. Is that it primarily reactivates the hedonic aspects of the unconditioned stimulus. Okay, so cool. But again, it's all about the methods. So just to summarize kind of what we were able to do. So again, we wanted to know, does the pattern of activity when they experience pain or brush come to predict the pattern of activity when they see the face that's associated with the tactile experience? And yes, it does. And what modeled information contributes most? And then in prefrontal cortex, mostly emotional information, but this is just another sort of image of what we're able to say, sort of look at in this case, the VMPFC. So this is actually the similarity matrix we observed. We were able to find the combination of patterns of interest, idealize similarity matrices that actually predict the data and reconstruct the data really nicely. And then using, again, regression in a second level, we were able to predict the degree to which each of these patterns of interest actually contributed to this overall pattern. So this is kind of a nice tool to be able to model what's being instantiated or represented by these brain regions in these contexts. So the next thing we want to do is kind of pull it into our translational study, and we got a Kickstart grant to get some pilot data, but we didn't get a big enough Kickstart grant to get enough data, because these days that's a lot. So this is, again, just a reminder of the current translational project. And I'm just going to show you a little bit of preliminary stuff that Brandon has been working on, but again, I'd love to spitball, like, what could we do next? He's got one perfect fMRI data set to play with, and yeah, with two tries at CIHR, no go, but you know, we'll keep trying.
Give me 6.
Wow. Okay. I don't know if that's encouraging or discouraging. The trouble is, by the time you get to round 6, all of the students and postdocs who were like generated the project are long gone, which is the heartbreaking part.
I'm really aware.
Yeah.
Oh, we've got a question in the chat.
Oh.
Really interesting results. How much of this do you think is dependent on the facial recognition processes? I do expect similar results to animals with object images compared with stimuli.
Yes. I do. I don't think that we actually use the faces. Like, maybe if we're looking at, like, if we had a region of interest that was the, you know, fusiform face area region, it would probably be more reflective of faces. But actually, when we looked at visual areas, we really looked at like a large hunk of the whole ventral stream. So I think it would generalize to different and also to just abstract shapes as well. So So this is a task where we want to look at representations of active and inhibitory avoidance behaviors as they are learned. So in this study, it's a study back translated from one of Stan Floresco's rodent studies, which was, I guess, forward translated from previously from a human study. But this is us trying to recapitulate. We tried to be much more literal about back translation. Stan really wanted people to be shocked in the foot. We're like, but the hand is like a forepaw. And we were doing it, but then COVID happened, so we had to use unpleasant noises instead for aversive stimuli. So basically, in this task, we have zillions of versions of it, instructed and uninstructed, but this is an uninstructed version that my student Ryan Tom designed. And basically, they in this task they have to learn that for active avoidance, they have to press a button a whole bunch of times in order to avoid a really, really unpleasant sound, worse than a shock. And for inhibitory avoidance, they learn that another cue means that they have to actually withhold a response in order to avoid that unpleasant consequences. And then there is a reward-seeking condition as well. So they learn a third cue. When they see it, means they have to press as much as they can in order to get a points towards a prize. And in the fourth condition, they have to learn that they have to withhold the button press in order to get the rewards. So what we had to start with in terms of brain predictions was Stan's papers with Patrick Pantadose and Julia Capuzzo, where Stan had found that using inactivation approaches that the rodent nucleus accumbens and the pre and infralimbic cortices play distinct roles in active relative to inhibitory avoidance and reward seeking, and that infralimbic cortex actually showed sort of mirror opposed patterns of opposed roles in relation to reward seeking and avoidance. So prelimbic and infralimbic cortex are sort of homologous to bits of the dorsal and ventral anterior cingulate cortex in humans, and they are hooked up to the nucleus accumbens. So at first we thought, okay, well, let's see what regions in humans are co-activated with the nucleus accumbens for active relative to inhibitory avoidance relative to reward seeking. This is the last data slide, by the way. So hang with me with this one, because there's a few layers. So this is a connectivity analysis that looks at differential patterns of activity for active relative to inhibitory avoidance, and this is one pilot subject, and reward seeking separately. So red is, make red be inhibitory. Yes, active is blue and red is inhibitory. And so actually can't see on my screen, so I'm going to go look at the screen. So you can certainly see that different medial prefrontal cortex regions are co-activated with the nucleus accumbens for active relative to inhibitory avoidance and reward seeking. So we are pulling up sort of like large scale, but sort of homologous regions to what we saw in the rodents. So that's encouraging. But we're also seeing, the nucleus accumbens is also co-activated, selectively co-activated with the nucleus accumbens for active relative inhibitory avoidance and reward seeking in these posterior cingulate regions and up in parietal regions. So so there's actually more going on here as well that one could explore. And then the other thing that Brandon did in the pilot data was to look at the degrees to which the active and inhibitory trials were correlated with each other. This is just avoidance here right now. Were correlated with each other over time with learning, because this is an uninstructed task. So at the beginning of it, they don't know they're learning what actions lead to what outcomes.
This would be sort of like you've either got to do something or not do something.
Exactly.
It doesn't matter whether avoiding punishment or getting.
So there's different cues. So there's a cue for active and a cue for inhibitory avoidance, and same for reward seeking. So there's separate cues.
But we're looking to see how the active response trials are similar to each other.
Are similar to, so in this case, this is the similarity between, now this is similarity between voxel patterns. So this is correlations on the y-axis, and it's basically these dots is like one square in one of those similarity matrices. So it's a correlation. And this is between active and inhibitory. This bottom plot is just avoidance trials, just to pull them, just to reduce dimensionality. This is the degree to which active and inhibitory avoidance trial 1 are correlated. In the dorsal HCC, that's blue, and the ventral ACC is orange. Okay, so this is with learning, the correlations between active and inhibitory avoidance are getting smaller, which means they're learning to discriminate active from inhibitory avoidance trials. So over time, they're starting to pull apart, and then less. It's 2 pilot subjects. What do you want?
Yeah.
But for a while. Yes, for a while they were sort of starting to disambiguate them. And what may be happening here actually is, so at first they always have these forced trials where they're given the consequences to make sure we know what it looks like when they get the consequences, and they know what the consequences are. So they may be actually representing the consequences less and less as they learn, and so that may be what's going on there. But that's something we can look at in the future. So basically, all we can see with that is they're discriminating active and inhibitory avoidance more over time up to a point. Okay, but then what do we do from here? Like, so what is an interesting way we could use the POI approach? So the first thing that sort of Brandon and I thought, we need to do a lot more brainstorming, and that's why it's fun to talk to people about. So what information about the effort versus the rewarding or aversiveness of the outcome is instantiated in the brain activation? And they could be instantiating information about like the amount of effort. Do I have to press or not press? The consequences of the error? Do they recapitulate the pattern of activation that we see when they actually get the unpleasant or pleasant outcome? Or the consequences of a correct response, which is more the safety response. It's more like in the previous study, those little bits of the grid where the CS minus bits of the grid. So that might be interesting to see just in a sort of relatively healthy population of controls. Yeah.
In the previous graph, was those correlations, were they when just the cue was presented? Or
Yes, thank you. Yeah, no, so that is just when the cue is presented,
okay.
But we also have them for the outcome.
Yeah.
So we can really reconstruct the way to which seeing the cue recapitulates the outcome or different aspects of the outcome.
Really,
yeah.
Really cool. Yeah.
That will be fun. And we had preliminary pilot versions of the task where reward seeking and where the reward and the aversive were separate, but the task actually works better because humans learn so fast, and even sometimes rats when they're intermixed.
Right.
So we're now able to sort of look at the sort of one pilot subject with the sort of optimal version of the task, where we can compare the reward trials with directly compare the reward trials with the aversive avoidance trials. So then, so then, like, once we also in our CIHR grant, we want to look at a clinical population with major depression. And so one thing, so in depression, like, what kind of information then are we more likely to instantiate? Might it be more about like the effort in reward seeking? Might it be more like recapitulating the effects of error in avoidance? Is there a pattern that's unique to people who are high in both anxiety and depression to compare to somebody who is high in one or the other, or high in neither? Obviously that would require a bit of a sample size to look at, but this method is pretty, we can look, it's a slow event related design, so we can look at the patterns of activation to single trials. So we do have a certain amount of power to look at what's going on in sort of smaller groups of people. And what would be really interesting to see is like does the pattern of the information that's represented change over treatment, for example, successful treatment for major depression. So those are kind of places we could take it. I see there's chat questions. Oop,
Okay.
it's all you. Oh, okay, right. Susan couldn't hear. Okay. So yeah, that's the kind of thing. It's like, okay, now we've got this tool, and this is sort of the first things we've thought of that we can use it. But what is another way that we can use it, or what are other ways or elaborations of way that could be useful in translational research? And before we get chatting, I want to thank so the real key collaborators who did the work on this study were Jamie, Mana, and Brandon, and then Adam Anderson was a collaborator at Cornell. We collected all the Pavlovian data at Cornell, and my lab, and their farm implements. Can you see how happy we are post-COVID? That was this summer. We finally got to be in the same place. Yeah, so ideas, questions?
So, no one on Zoom's gonna be able to, but,
If you yeah, let me mute myself. I'm gonna mute and you.
I think somebody has to unmute. Sorry.
Sorry, I probably just, oh, now I can hear myself in the room.
Down your staff. You don't have to turn off your phone. Right.
Yeah, no, okay. Kind of.
I think you just have to turn off your speaker if it's on. Just
It is
oh.
off. I can't.
Maybe Beck, if you're in the same room, you can just unmute yourself and go over.
Okay, I'm unmuted now. So that works. All right, now you just need to speak up.
now I just need to yell. Well, so the nice thing about this is that you're gonna be able to infer what representations are being used during learning, I guess. So like, you know, this is a really good example of the Pavlovian conditioning example was really clear. So I guess this is going to be, I'm just stating the obvious really, this is going to be the most useful when it's ambiguous what representations our subjects are relying on.
Exactly,
Right?
exactly. If it's obvious, there's no need.
Yeah.
And it's the representations of which brain region. So it might be obvious, like certain big hunks of cortical real estate or subcortical real estate may be representing different information.
Right. And right, so then we can disambiguate which brain regions are sort of representing what. So,
Yes.
just like you did in the Pavlovian task.
Yes.
ventromedial prefrontal cortex is representing valence, and
Yes.
somatosensory cortex is representing the kind of touch, and it doesn't matter really what the valence is.
Yeah, and for example, so we could actually like we could even like choose like hunks that are shown to be co-activated with the nucleus accumbens and make them regions of interest and say, okay, what is this little particular chunk that's co-activated with nucleus accumbens representing?
Right. So can I ask Luke a question? Maybe he can't hear me. Maybe I'll just.
Luke, can you hear Kath?
I can hear.
So Luke, when you looked at the similarity between the response to near misses and wins a long time ago in a neuron paper, did you use something like RSA?
No, we didn't, but that would be quite a good idea. In the neuron paper, we compared wins against both kinds of non-win outcomes, full misses and near misses, and then we broke the baseline outcome type in half to compare near misses against full misses. But yeah, I was also thinking, I'm sort of interested in the idea of kind of mixed emotions at a very colloquial level, that there are certain events that we experience that are that have kind of good and bad elements, and near misses being one of those, that they seem to be subjectively unpleasant but quite motivational, and whether RSA could pull out that kind of signal.
I think so. I think it could, and one of the things I like about RSA is it's kind of agnostic to the medium that it comes from, right? All you pull out is a correlation matrix. So you can use it with EEG or psychophysiological data. You don't even need to use fMRI scanner unless you mostly care about the brain region that's showing the representational pattern. If you just care as like, are people's physiological responses or cortical responses showing representing certain information, then you could use it with EEG. People use RSA with EEG.
So you could use RSA more easily, with the classic like contrast fold approach that Luke is talking about. With RSA, you could say in a gambling task, which I guess is what I'm most used to thinking about recently, you could look at a loss outcome and you could infer that that's like a negative emotional state. I mean, that's an assumption. And then you could see when a sort of risky bet cue is presented, whether there are brain regions that are treating that or representing the negative emotional state or active as if they were going to get punished at that time. And you can't really do that with contrast analysis, can you, in the same way?
No, you can't model, like you can contrast experimental conditions, but all of the information has to be in the experimental conditions. So this is kind of a way, like, as you know, very different from what computational modeling does, but it is a way of using modeling to sort of pull out information that's sort of more nuanced than is in the actual experimental manipulation. So, and it's interesting, based on sort of what I'm seeing in others' research, often like in a case like a near miss, you might have like one brain system representing the rewardingness of it, and others representing the loss, and the different people, right? There might be a push-pull, and different others might win out. And I've seen people use not RSA, but multivariate pattern analysis. I'm totally spacing on his name. It'll come back to me, but to sort of look at these kind of push me, pull you sort of effects between reward and punishment, or between sort of reward and neutral in these kind of tasks where there's multiple things going on, which is usually right.
Yeah, I mean, that's the thing. I'm wondering if, I mean, I don't think you would, but would you need to have like an explicitly negative event as well? So, you know, if you had a hypothesis that, you know, the loss of money was not as impactful to a subgroup of individuals, whatever,
Mhm.
they're just not, maybe not paying attention to the losses in the same way. So the loss outcomes aren't represented critically between subjects.
Mhm.
Would you need to have something that was like, this is a really nasty thing? I may be playing your nasty noise,
Yeah,
and it's like, that's unpleasant.
that would probably be the most reliable
Yeah.
way to do it.
And then see to what degree
Yeah, you
a loss
recapitulated that.
stimulus.
Yeah.
It would just be really interesting to do that.
You could potentially just use like totally ideal patterns of interest if you could sort of articulate or visualize what the ideal voxel pattern would be with a really unpleasant event. So for example, Jamie's valence POI, or his all pleasure, all pain POIs, right? He actually didn't need the actual, the first proof of concept was to see the degree to which they contributed to the actual activation when they were experiencing the event. But actually, when he was using it with the US with the touch stimuli, he actually didn't. He wasn't. It was like it was all a priori. This is what it would look like. So you could conceivably do it a priori and just say, okay, this is what a really negative event would look like. This is what the similarity matrix would look like, and use that. It's nice, though, I think, to also have the step where you actually show that indeed that is what happens when you have a really negative event. I think it's more convincing that way. And there was an interesting conversation. So both Brandon and Jackie from my lab are at the brain connectivity conference. Well, they're remotely at the Whistler Brain Connectivity Conference, which was to go in person, although Whistler is close, it was like $10,000 a person. Like a ski vacation for the well-funded, but they opened a remote option, which was lovely. So they were there, and they were just talking about the difference between like. So Jackie really wants to find neuromarkers, right? She wants to find patterns of activation that discriminate, somebody who has one condition from somebody who doesn't. And so she's really looking at patterns of brain connectivity, sort of creating brain scores out of that, which I think is really useful. But Brandon was asking, so is this RSA approach good for finding neuromarkers? And so that was good because Brandon and I were able to have a conversation where I said, I don't think that's what we're doing here. I don't think that's our goal. And I don't think that's what it's good for. I do think brain scores using, connectivity, the large connectivity matrices are probably a good way of finding neuromarkers. This is it's a little bit more subtle. If you want to look at the information, like in exactly the context you've been taking is represented. If you want to see how sort of information that's represented in these brain systems changes, with treatment. And that's what that's for. It's not about finding, here's a signature of, major depression, or here's a signature of when you recover from major depression. It's much more like, here's what your brain may be representing more in major depression relative to other things. And here's how that might slowly or not slowly or quickly or not at all change with successful treatment. And I think that's a different kind of information.
Yes, we've got a couple of questions in the chat.
Oh, okay.
So Daniel asks, I don't know much about fMRI data analysis, but is it possible to use manifold learning concepts to see how the activation patterns change from resting state all the way to learning? Is that possible, or are the data too slow on that?
I don't know.
Good answer. Next question.
Brandon might know. That's a question to fire at Brandon.
Right.
He could probably answer it.
And then I think this is exactly what we've been talking about. This is, would a similarity matrix get at the degree of aversiveness? If you think of a loss win in terms of expected value ratio, would that be more or less neural representation?
Yeah, that's what RSA is really good for, as opposed to just using a sort of multivariate pattern analysis, MVPA machine learning classifier, right, which discriminates one thing from another thing. That's what RSA really gives you, is the degree of similarity or dissimilarity. So it's nice for that kind of question.
Yeah, because can you get a math? Oh yeah, you can with that Euclidean distance.
Yep.
Thingadoo.
Yep. And so Brandon's been playing around with a lot of that lately on another sort of study that he's working on, just representing Euclidean distance. So he's developing some chops at doing that.
Yeah, so then you like theoretically know, or at least, yeah, whatever, whether, you know, when people are in a negative mood state or experiencing depression versus depression and anxiety versus just anxiety, whether they might be representing certain contexts as more negative than others, or certain stimuli as more
Yeah,
negative.
is more negative, or yeah, and even what aspects of it are more negative? Is it the effort, for example, or is it the unpleasant outcome?
Right. Yes.
Yeah.
Yes.
Yeah.
That's really cool.
Yeah. Go Jamie. Oh, he's
What a
going.
great lad.
He's going to Thunder Bay. I know. You'll miss him.
That's really cool. I need to remember all this. Because I've had my computer on, I haven't been my computer. I haven't been taking any notes.
Oh, we've been recording.
Yes.
I'm gonna stop recording though now, I think.
Shut up.
I think now would be a good time to stop recording. It's just gonna get too